Hello, my name is Jadzia Evans, and today we're going to be talking about a new species of Trichinella. Understanding parasitic diversity and distribution is essential in managing the potential impact of parasitic diseases in animals and people. Imperfect diagnostic methods may conceal cryptic species. Today we're going to be talking about the discovery of a previously unrecognized species of Trichinella in wolverine from northwestern Canada that was indistinguishable from T. nativa. It was discovered when sequencing a mitochondrial genome. It was discovered accidentally when sequencing the genome of T. nativa. Of 95 individual hosts from 12 species of mammalian carnivores from northwestern Canada, from which larvae were identified as T. nativa, only wolverines were infected with this new type called T13. These infections were single or mixed with T. nativa and or T6. That brings me to my first slide. This is a map of the Trichinella that they found. The blue one represents T. nativa, which is the one that they had previously already known that was existing. The red one is the new type of Trichinella that they found almost in the same areas, and it comes from a wolverine, which is pictured on the bottom right, and an image of the parasite is on the top right. Now, Trichinella is a parasite that you can get from eating raw meat or undercooked meat, and animals can get this as well as humans, and it can cause diarrhea, nausea, stomach cramps, loss of appetite, and fatigue. Next, we're going to talk about the methods and materials of how they found this new Trichinella strain or species. They did this by using larvae. Larvae of Trichinella were recovered from previously frozen wolverine muscle tissue by the double separatory funnel digestion method. Now, this is a method where they take some filtered digestive juices from a dead wolverine and leave them in a separatory funnel for 30 minutes to a few hours. After a while, the larva will settle at the bottom, if there is any at all. DNA was extracted from pools of 15 larvae from an individual wolverine using Promega DNA IQ kits. This is a separatory funnel. Visual examination and motility testing confirmed that T13 is encapsulated and likely freeze tolerant. After motility testing, they ran a few genome tests. They did this by using a PCR restriction fragment length polymorphism assay. In other words, they used restrictive enzymes to cut down the DNA molecules. That brings me to slide 3. The last 3 lanes, such as T2, T6, and 13 on the slide, show restriction fragments resulting from an amplification and digestion of T. nativa, which was T2, Trichinella T6, and the newly recognized species, which is called Trichinella chanchalensis, also known as T13. Lanes 2 through 5 on the slide are the new species of Trichinella, also known as T13, and those in lanes 6 and 8 through 12 are T. nativa. They proposed Trichinella chanchalensis for T13 based on significant genetic divergence from other species of Trichinella and broad-based sampling of the Trichinella genome. Species of this cryptic taxon were initially identified as T. nativa based on the location of bands on a conventional PCR used for identification of isolates within the genus. But because of this research, they found out that it was a completely new species of Trichinella. Exploration of Alaskan and Siberian isolates may contribute to further resolution of a complex history for species of Trichinella across Beringia, including Trichinella chanchalensis. They discovered a new species that was previously unknown. Now that the discovery of this new species of Trichinella has been made public, the CDC can begin researching this new species. With this new research, the CDC can watch out for any outbreaks of the new species and find a way to avoid any outbreaks in the future. They are now able to find a treatment for this new species because of this discovery. I do agree with the conclusion in this research, and I liked this article a lot. Thank you.